for the course. And typically the way lecture will be recorded is by echo in the classroom. And unfortunately, today, first run through, among the other technical glitches I had, the echo recording doesn't have an audio track for some reason. Hopefully I'll get that fixed by Thursday. But just so that people that missed class or are in the online section know what we went over, they can have this audio, which may not be as comprehensive. I'll try to make it as brief as possible, but at least you have all the information, and I'll try to highlight the really important stuff. For the most part, just going to cover typical day one kinds of things. I'll introduce myself, talk about the syllabus and Canvas resources, just a little bit about why take bioinformatics, and then look ahead specifically at what is due next Monday and what we're going to do the rest of the week, which is Thursday. Before I get to what is bioinformatics, let's go ahead and jump over to the syllabus. So this is the Canvas shell. If you want to scroll down, right, day one, this is basically what we're doing. You can get the syllabus there as a Word file. I jump over to that. Okay. So here, this is me. My office is in ERB 430. Got some links to my UTA profile and my lab website. I'll look at those a little bit more in a minute. And office hours are 11 a.m. to 1 p.m. on Tuesday, Thursday. So basically right after the in-person section meets. If you can't make it at one of those times, feel free to reach out and set up an appointment with me. Best ways to get in touch with me are email or Teams. Just send me a chat in Teams. If I don't get back to you within a day or so, feel free to reach out again. You probably just fell off my inbox page. Don't feel like you're bugging me. Sometimes I just have too many streams of communication coming through. So there are 3 sections in the course. 2 are in person. Those are the ones that are meeting in Pickard Hall on Tuesday and Thursday from 9:30 to 10:50. The recordings of those videos will normally go live shortly after class meets on Tuesday and Thursday. So the asynchronous online section, which is the 3rd section, has access to exactly the same information. All the course material is the same for all 3 sections, which is comprised of primarily biology and undergraduates and graduate students, and then master's students in data science and undergraduates in data science, with a few other PhD in bioengineering, some undergrads in bioengineering also mixed in. And occasionally we get majors from other departments and colleges. So I have some information here about the room capacity, and this is mainly because since we're all covering the same material, if you're in the online section, you are welcome to come attend the in-person, as long as there are seats available. So right now there are about 12 open seats. The room was pretty crowded this morning, but that tends to thin out, and I'll get to why that is in a minute. As the semester goes on, because this is project-based and people will be able to work ahead so they don't need to be in class. Plus, everything is recorded and posted on the website, so it makes the in-class somewhat less important, although I encourage anybody who's able to come. Just know that if you're in the online section, that the priority has to go to those in-person section people. And so it might be better to just reach out to me to ask if there are or have been open seats before you come. That way you have a little bit more information to go on. You're welcome to record, but like I said, Echo is going to be recording, and hopefully we'll have the audio fixed by Thursday. Some student learning outcomes. I'll let you look at those on your own. There is no textbook, and technology requirements. Basically, you need a semi-recent laptop. If you can run the latest versions of Windows OS or Mac OS, you will be fine. If you run Linux, also fine. Most of our computing, especially the heavy lifting, will be on remote servers, Google Colab, and some of the NSF-supported infrastructure in Pittsburgh, which I'll get to again in a minute. Assignments, there's weekly homework. There's a detailed schedule at the bottom of the syllabus. And then the last month or so of class is comprised of final projects, which can be individual or group, small group. Lots more details will come on that, but the grading for the final project falls into sort of 4 discrete assignments. You'll have a proposal, then a draft report, which is like a summary. Then we'll do have a peer review of that progress, and then you'll have a final report with a presentation due on the very last day of class. Grading information, this is just a breakdown of the assignments at a very high level, just so that you have an idea in proportion to all the other assignments in the class, how much each one is worth. Often I just make them worth 10 points, but that's just so that it's easy to translate into a percentage scale. And just know that at the end of the semester, those will be weighted by the values in this table. So for example, the 2 assignments that are going to be due next Monday are combined worth about 6% of the grade. Pretty standard grading breakdown. Don't anticipate needing to have a curve. I've never had to implement one in the past. The late work policy, deadlines are hard deadlines. Just know that going in. If you miss the deadline, you can still turn it in for 50% credit up to a week after the due date. After that week is over, you get a 0. That applies to everything except for the final reports and presentations, which, because they're due on the last day of class, they're just going to get a 10% penalty per day after the due date. There are no exams in the course, so don't need a makeup exam policy. There is an opportunity for extra credit when we get to, scroll back up here, this machine learning section. There's an option to do a single-cell RNA-seq or a metagenomics assignment. And if you do both of them, you have the opportunity to get a little bit of extra credit. That could make up the difference in say, you had a late assignment earlier in the semester. Could certainly make up the difference for that. The rest of this are course and university policies, basically boilerplate things that you'll see in many of your syllabi for the semester. Hopefully, grade grievance policies and stuff like that won't be applicable, but they're here if you need them. And then, like I said, at the bottom here, there's a course schedule that just gives you an idea for the module number and then the dates that we're covering those modules. You'll see basically 1 module per week until we get to the end of October. And then the last month of class is set aside for working on projects. So that's the syllabus in a nutshell. I'm going to jump over to the Canvas shell. So after the syllabus there on day 1, next thing would be to go to the Getting Started module. These top three are very specific to the course. The bottom three are, again, just like boilerplate stuff in the syllabus, typical for most of the courses at UTA. So first thing is in getting started, who am I? This is me. I'm an associate professor in biology. You could click there to look at my UTA profile. Uh, a better way to see what my lab does is to check out Google Scholar. You can see, hopefully many of you are familiar, if not most of you are familiar with Google Scholar. These are my publications. Um, if you want to know what any scientist is sort of recognized for in the scientific community, you can look at what papers get cited the most. So for me, it is um, computational research. So all this top group are either genomes that I've been involved in, genome sequencing projects, or that top one, Cafe, is computational software that I developed while I was doing my postdoctoral research to be able to infer the evolutionary forces acting on gene duplication and loss. So how genomes gain and lose genes and why, basically. Then you can also look at the lab website, which is constantly under construction, even more so since the summer. Um, uh, One of my favorite quotes there at the top, and then some AI generated art just showing beetles have an amazing diversity. Haldane is one of the fathers of the modern synthesis in biology, and he had a funny quote that if one could conclude as the nature of a creator from a study of creation, it would appear that God has an inordinate fondness for stars and beetles because there are just so many of them. Uh, what does the research in my lab actually look like? It's broken down into three broad categories. This horn beetle system, where we're studying the genetic basis of conflict. These guys are really interesting. They have horns, and the males will sort of sumo wrestle each other until one submits, and then they will just stop trying. They basically stop moving around very much. They won't try to mate anymore. And so we are studying the molecular basis of that behavior. We also study sex chromosomes. Sort of a foundational question there is, how do males get away with having only one copy of all those genes on the X chromosome? Um, and I suppose you could flip it and say, how do females get away with not having any of the genes on the Y chromosome? Um, if you have an autosome that is that way, more often than not, it's lethal. And yet for sex chromosomes, that's the normal state. And then we still are involved in lots of comparative genomics projects of all kinds of organisms. For both of those first two, it tends to be beetles as our main study system, at least the experimental study system. Bioinformatics and theory questions sort of bridge into other organisms. But then when we get to comparative genomics, we do some beetles, but I've also worked on plants, all the way from plants to fungi to um, macaques and chimps and humans. People in the lab. These are the current grad students. Shanna just graduated, and we have two new students joining two PhD students joining the lab. Some of the undergrads that are working in the lab. If you're interested in joining the lab, there is some opportunities. The first step, though, would be to do well in this class. And then publications you can sort of sort through and look based on topics and things like that. If you're interested to dig in more to what my lab does. I already talked about the contact info. It's in the syllabus, and then office hours. This other stuff is also in the syllabus, just laid out a little bit differently. Again, you've got the student learning outcomes there. And the required materials. That this stuff is all duplicated just laid out differently from the syllabus. It reminds me though that back on the home page, I didn't scroll down all the way, that the home page does have a schedule that lays out the due dates for all of the assignments. That's all I need to cover in the Getting Started module. There is a page on tech requirements that has a little bit more detail than what's in the syllabus. Whoops. Don't know what happened to my link there. Ah, there we go. Nothing big there on the hardware requirements. Like I said, if you have any modern laptop, it should work. Um, just this gives some specs to give you ballpark. But if you're running modern operating system, you should be fine. Um, software requirements. This is we're going to have to set up, or you will have to set up accounts on several different platforms. Gives you an idea of some of the software tools. Some of you may be familiar with these, and some may not. That's fine. We will deal with them as we get to them. Just wanted to show you what that stuff was. The rest of the Getting Started module is general for UTA students. Third thing that we're going to hit on, in terms of Canvas resources, is jumping right into this module for module 1. So proceed to module 1. It's a little bit of an overview about what gets covered in module 1. We're going to pick up on high performance computing, do an intro on Thursday. The learning outcomes here, basically what we're doing right now, covering the syllabus and understanding the sort of course layout. Getting you registered on Bridges 2, which is I'm going to show you in a second. And then there are some videos to look at, and you'll have a reflection on AI use as part of your assignments for this week's material. And then you've got a brief to-do list. Watch the lecture video, which you're watching at the moment. Watch the videos for a history of generative AI and the Sam Altman interview, and then register for access and log into Bridges 2. So if I hit the next button, it'll take me. This is the main page in every module that you're going to interact with. There will always be a Tuesday and a Thursday lecture slot. This is the Tuesday lecture. Oops, it's reloading. You can download the PowerPoints, so if you clicked that link and downloaded it, you would get this Welcome to Bioinformatics PowerPoint. This is just another place linking you to the AI reflection assignment videos. I'll get to that in a second. And then typically you're going to have your video recording right here. Now, this is the echo recording, which usually is all that will be there. But as it says, there's no audio track for this echo. So if you open this up later, if you're actually watching this, there are 2 of them for Tuesday's lecture, because I'm going to upload this as a second lecture link. Okay. Thursday, right? After Thursday, hopefully there'll just be one, and it'll just be the echo recording. It'll have its own PowerPoint that goes along with it, the intro to HPC. And then here you've got a link to the account creation instructions. Now, those account creation instructions also appear in the PowerPoint slides. So, right, I can download that. It's my intro to HPC, right? So this is the lecture that I will do on Thursday, and it has step-by-step how to create your Access and Pittsburgh supercomputing accounts. Okay. Now, all that information that's in that lecture is sort of condensed into just the steps you need to do on this page. Okay. Just noticing there are several Canvas glitches here that I need to iron out. You probably won't see the Google Colab, Rosalind, and GitHub links there. We're going to set those up later. And just to simplify things, I'm just having you focus on Access and PSC to start with. Okay. So that's the lecture content. Tuesday's lecture content is going on. Somehow got launched into the fall 25 version of this course. Hmm. So one of those links is broken. So here is what it should look like for you. Right, where it's just showing the Access PSC link. This is all correct. This is the videos for the reflection assignment. If you go into modules, you can always navigate straight to things. Hopefully you're familiar with how Canvas operates. But so those account creation instructions I was talking about, we'll cover that in more detail on Thursday. The videos for the reflection assignment are here. There's two of them. And then your assignments are the PSC login quiz. So once you've completed everything in the Thursday lecture, you should be able to log into the Pittsburgh supercomputer. And what you're going to do is give me a screenshot showing that you are actually logged in. So that's one of the assignments for next Monday. And then the other assignment is the reflection on AI quiz. Well, quiz. It's just a reflection, 400, 600 words, and you answer those 4 questions. Okay. After you've watched the AI videos. Okay. I think that about covers it. I don't want to spend too much more time. Hopefully all of the technical glitches will be out of the way, and on Thursday, everyone will be back to having exactly the same information. Okay. So I'm going to go ahead. Record.